IB Bio students. Today we're going to continue 8.1 and we are going to study enzyme inhibition. So the essential understanding here is that enzyme inhibitors can be either competitive or non-competitive. So we're going to look at the two types of enzyme inhibition pathways. So what is an enzyme inhibitor? Well, this is a molecule that disrupts the normal reaction pathway between an enzyme and a substrate. So in other words, it's going to prevent the formation of an enzyme substrate complex in some way. Okay? So we said that the enzyme inhibitors can be either competitive or non-competitive. So we're going to see how they work in just a moment. But before we do so, let's look a little bit at a normal enzymatic reaction. So this is how an enzyme normally works, right? So in a normal reaction, here we see the substrate, and here we see the enzyme, and we see how the substrate binds to the enzyme on the active site of the enzyme. Remember that the active site is found on the enzyme, and it forms an enzyme substrate complex. Okay, so we know that the substrate shape and the enzyme shape are somewhat complementary, right? And that's what we call enzyme substrate specificity. And we know that when the binding occurs, there is a conformational change of the active site, allowing the substrate to fit in, right? And remember the example that I gave you is like trying to squeeze into a shoe that's not quite your size, right? That's too small. So that's what we call an induced fit. That means that the active site of the enzyme is going to undergo a slight conformational shape so that the substrate actually fits. Okay, now that conformational shape of the active site is what's going to destabilize the chemical bonds within the substrate, and it's going to lower the activation energy, allowing for that substrate to break up into products. So here we're seeing a catabolic reaction, and we see how the product formation here is actually accelerated. Okay, when the products are formed, the enzyme is released, and the reaction can happen again. So one thing to note here is that you're all aware here of this active site of that enzyme. So we call that the active site, correct? But remember that we've talked about how enzymes have another site called an allosteric site. So this site is going to become important in just a moment, okay? All right, so let's move on. So what happens in competitive inhibition? And let's look at that one first. So in competitive inhibition, you have another molecule other than the substrate. So in this case, it's this molecule right here, and we're going to call that the inhibitor. So in competitive inhibition, this inhibitor is going to bind to the enzyme's active site. So it's going to prevent the substrate here from binding. So it kind of kicks it out. So the competitive inhibitor is going to block the active site, so then you get no enzyme substrate complex. Instead, you get an enzyme inhibitor complex. And that means that there is going to be a blockage of the reaction. The reaction is not going to take place, and as a result of the inhibitor. Okay. Now, something important here, and we're going to look at this further when we start our activity. It says here, let me make my pen smaller here. It says here, as the inhibitor is in competition with the substrate, the effects can be reduced. So this is saying that the inhibitor effect can be reduced if you increase the substrate concentration. So if you start adding more substrate, right, because you keep the same amount of enzyme in the reaction, that means that there's a greater chance for this substrate to bind to this enzyme. So if you increase the substrate concentration, even though you have an inhibitor there, right, that is going to increase the chances for this substrate to bind with that enzyme. Okay, so let's look at a curve here. So in this curve, we are going to label the substrate concentration here at the bottom. And we are going to label the reaction rate here on the y-axis. And we're going to call this the Vmax, which is the faster or the most pronounced amount of substrate enzyme concentration that the reaction can have. So if you have an uninhibited enzyme, right, and you learned this in HL, if you have an uninhibited enzyme, and let's label that, uninhibited enzyme, as you increase the substrate concentration, right, so first the reaction is going to speed up, right? You have enzyme substrate complex, the reaction is going to speed up, but eventually you're going to reach a max. That means that even if you keep adding substrate, there's just going to be a certain amount of enzymes to catalyze the reaction. So then the reaction is going to flatten out. Okay? Now what happens when you add a competitive inhibitor? So I'm going to use a little dashed line to show you what happens. So when you add a competitive inhibitor, look at what's happening here. And I'm going to label that here, competitive inhibitor. So if you add the competitive inhibitor, if you increase the amount of substrate, what is going to happen? So this is what I was saying just now. As the inhibitor is in competition with the substrate, its effects can be reduced by increasing the substrate concentration. So if I increase the substrate concentration in the presence of an enzyme inhibitor, eventually I will get to reach Vmax, right? Because if I add more of this substrate here, all of that substrate that I've added, there's a greater chance for that substrate to form a enzyme substrate complex, right? Even if an inhibitor is there. So even with the presence of inhibitors, right, as I increase substrate concentration, this reaction can eventually reach Vmax. Okay? So that's the only way to counter the effects of an inhibitor, by increasing the substrate concentration. Okay? All right. Now let's look at a non-competitive inhibition process. So in a non-competitive inhibition process, here we have, again, the enzyme, the substrate, and the inhibitor. But this time, the inhibitor, notice it's binding to the active site, the allosteric site of the enzyme. So the inhibition does not happen on the active site. The inhibition happens in the allosteric site. But once this inhibitor binds to the allosteric site of this enzyme, what ends up happening is that there is a conformational shape of the active site. So look at what's happening to the active site. It used to look like this. Now it looks like this. So then that means that the substrate cannot bind. So if the substrate can't bind, then you can't have the reaction take place. And that's it. The reaction stops, right? Now let's look at this part right here because this is important. Now, as the inhibitor is not in direct competition with the substrate, increasing the substrate level cannot mitigate the inhibitor's effect. So if we draw our graph again, and again, let's label this substrate concentration. Let's label the y-axis the rate of the reaction. And let's call this the Vmax, or the maximum rate of the reaction. In an uninhibited enzyme, as I increase the substrate concentration, I am going to increase the rate of the reaction until I plateau, right? So I can keep adding the substrate but eventually I'll reach a Vmax because I have a certain number of enzymes, and that reaction is going to plateau. So this is what happens with an uninhibited enzyme. But if I add a non-competitive inhibitor, right, since the non-competitive inhibitor is not directly in competition with the substrate, right, even if I keep adding more and more substrate, that is not going to mitigate the effect of the inhibitor. So that means that even if I add more substrate, my Vmax is going to be lowered, or the rate of my reaction is going to be lowered, okay? Because the inhibitor is actually affecting, on the allosteric site, it's affecting the active site of the enzyme. So I can keep adding as much substrate as I want, but it doesn't matter because all of the enzymes present in that reaction, they are going to have an altered active site. So then the reaction is not going to take place and the Vmax is going to be lowered. Okay? All right, so let's continue. So let's look at some real life examples of inhibition. So enzyme inhibitors can be used for a variety of purposes. So a lot of the medicines out there are types of enzyme inhibitors, and they actually treat a disease. So sometimes it's a good thing, right, to add a competitive inhibitor. And they can also be used in agriculture as pesticides. So the two types of examples that we're going to look at today, we're going to look at an example of a competitive inhibitor called Relenza, and this is the trademark name. And what Relenza is, it's an influenza neuraminidase inhibitor. And we'll go over that in just a second. And another example of a non-competitive inhibitor would be cyanide. And I'll show you how that prevents aerobic respiration in just a second. All right, let's put on our thinking hats then. Now, in the activity that you're going to work on, you're going to see other examples of competitive inhibitors and non-competitive inhibitors. All right, so here we have Relenza, a competitive inhibitor, and it's a synthetic drug, and it was designed by an Australian scientist, and it was used to treat the influenza virus. So in this case, we know that the influenza virus is the bad guy, right? So the Relenza, or the competitive inhibitor, well, that's the good guy, right? So let's see how the Relenza is working here. So first, let's try to understand this diagram here. So here we have a virus, and this is the influenza virus right here. So I'm going to label that first. And within that virus, we have many different things. So one of the things that we have, these little hands, is an enzyme. And the enzyme has a name. The enzyme is called neuraminidase. Okay, remember, everything that ends at ase is usually an enzyme. So this enzyme belongs to this virus. Now, in addition to having an enzyme, this virus also has a docking protein called hemagglutinin. So this is a docking protein. So you don't have to go into the whole biochemistry of how this works. But neuraminidase, what it does, or the enzyme, what it does under normal conditions, is that it cleaves or it cuts the hemagglutinin, or this docking protein. So when this docking protein is cleaved, right, then that allows for this virus right here to dock with an infected cell. So this could be an infected cell in your body. Now, if it's an infected cell, that means there's a lot of other viruses in there that have been replicating. Okay? So remember that we've always said that infected cells lyse or burst, right, and release a whole bunch of the viruses, but we've never really gone into how it does that. So today we are. Okay? So the virus docks with the infected cell, by allowing one of its enzymes, neuraminidase, to cleave a docking protein found on the virus. Okay? So once the virus is able to dock with the infected cell, then that infected cell is going to release all of the viruses, and now these viruses are free to go and infect other cells. Now, the infection mechanism works a little bit different than the lysing mechanism. So what you're seeing here is the lysing mechanism of the cell. Okay? All right, so that's how influenza works. So now let's look at how Relenza works. So Relenza is a competitive inhibitor. So if this is the enzyme that's responsible for the docking process, what Relenza does is Relenza is going to competitively block the active site of neuraminidase. So that means that neuraminidase can't cleave the docking protein. Therefore, the virus can't dock onto the infected cell, and therefore the other viruses that are inside the infected cells, they can't be released. Therefore, you can no longer spread the influenza virus. Okay? So in this case, this competitive inhibitor here, this medicine, is acting positively. Okay? All right. Moving on then. So let's look at an example of a non-competitive inhibitor. And in this case, this non-competitive inhibitor is acting negatively. Now, sometimes non-competitive inhibitors can act positively, right? And sometimes competitive inhibitors can also act negatively. So don't think that all non-competitive inhibitors are bad and all competitive inhibitors are good. Okay, these are just two examples that we're going over. So first, let's look at the normal functioning of the electron transport chain. So if you remember, the electron transport chain was a third process within cellular respiration. Okay, and we're going to go over this process in detail, but just quickly, right? If we have the outside of a cell here, and we have the inside of a cell here, we know that electrons move through these proteins and these molecules, also these enzymes. And while these electrons move from one protein to the other, this causes the release of hydrogen ions. Okay? So electrons shuffle around these proteins. Hydrogen ions are released into the intermembrane space. Now, these hydrogen ions, they come back in through a molecule here, or a protein called ATP synthase. And when these hydrogens come back in, that generates ATP. And then ATP is used by the cell for energy. Okay, so that's how you get energy in your cells. Cellular respiration gives you energy, right? Remember, 1 glucose molecules equals 36 molecules of ATP. In the process, you take the oxygen that you breathe in through cellular respiration, and that eventually turns into water. Okay, and the oxygen is the most electronegative acceptor here, and that's why these electrons are carried along the chain and pulled by the oxygen here. Okay, these guys right here are electron carriers, and we're going to learn more about them later. So this is how the electron chain works in like a nutshell here. Now, there happens to be a molecule here, or a carrier molecule here, that's part of the electron transfer chain, and we call this cytochrome C. This molecule is responsible for this whole process. Without it, the electron transfer chain wouldn't work. Without it, your cells would not gain energy. Without it, pretty much you would die. So then what is cyanide doing here? Cyanide, acting as a non-competitive inhibitor, what does it do? Well, eventually it prevents the ATP production via aerobic respiration and is going to lead to death. So what it does is it binds to the allosteric site of the cytochrome oxidase. So it's going to bind this protein here, or this carrier protein is actually an enzyme, and it does have an allosteric site. So cyanide is going to bind right here to an allosteric site of cytochrome oxidase or cytochrome C carrier molecule. And that is going to change the active site of cytochrome oxidase. So then that means that the electrons that are being passed on here through this chain can no longer pass and be received by the final electron acceptor here, which is oxygen. So the electron chain is not going to be able to happen. ATP is not going to be produced, and therefore aerobic respiration is not going to do what it's supposed to do, right? Which is take the oxygen to your cells and provide your cells with energy. So your cells without oxygen, your cells without energy pretty much means that you are dead. Okay, so in this case, cyanide is acting as a negative non-competitive inhibitor. Okay. All right. So I know that was a lot for today. If you need to watch this video again or pause it, you can always do that. And that's it for today.